Hello students, welcome to the EPG Pathshala. I am Dr. Pradeep Bere, Scientist, Dairy Microbiology Division, National Dairy Research Institute, Karnal, Haryana. Today I am going to talk role of biotechnology in food processing under the paper Advances in Food Science and Technology. Now let us come towards the introduction of this module. Biotechnology is the use of living systems and organisms to develop or make useful products or any technological applications that uses biological systems, living organisms or derivatives thereof to make or modify product or processes for specific use. This is the definition given by the CBD. There is many more applications of biotechnology have been given. This biotechnology as applied to food processing to make use of microbial inoculants to enhance properties such as the taste, aroma, shelf life, texture and nutritional value of the foods. By using various biotechnological methods or tools, we can improve the overall acceptability of the product. Fermentation is one of the very important area of the biotechnology. Fermentation processing is also widely applied in the production of microbial cultures, enzymes, flavors, fragrances, food additives, and a range of other high value added products. Biotechnological developments have resulted in the widespread availability of low-cost, rapid methods of identification when compared with the significant cost time requirements of the conventional techniques. One of the important aspect of biotechnology is the use of genetic engineering. Genetic engineering is the technique of removing, modifying, or adding genes to a DNA molecules in order to change the information it contains. By changing this information, the genetic engineering changes the type or the amount of proteins an organism is capable of producing. Now friends, let us come towards the learning objective of this module. In this module, the objective is to study food biotechnology, to study role of biotechnology in foods, to study various biotechnological methods to identify or diagnose potentially important microorganisms, to study the application of biotechnology in milk, food and dairy products. Coming to the application of biotechnology in dairy sector, so we can categorize how the biotechnology can be useful in the dairy industry. So we can categorize the use of biotechnology in two different ways: dairy production and processing. Under the dairy production, the applications includes recombinant bovine buffalo somatotrophin, recombinant vaccines, rumen manipulation, DNA fingerprinting, and RFLP, PCR-based diagnostics, especially in animal healthcare, embryo transfer technology, animal cloning, gene farming, and transgenics. In the processing, dairy processing especially, designing a milk through genetic engineering, genetically modified organisms, the starter cultures, mainly lactic acid bacteria, genetically modified foods, food grade biopreservatives, recombinant dairy enzymes of proteins, accelerated cheese ripening, probiotics, functional foods and nutraceuticals, dairy waste management and pollution control, gene probes and PCR based pathogen detections. So in the what it says, the biotechnological approaches can be employed for improving the productivity, economy, physicochemical and nutritional attributes of a wide range of food products. The genetic engineering is used in the production of drugs, human gene therapy, and the development of improved plants. For example, an insect protection gene BT has been inserted into several crops, corn, cotton, and potatoes, which give farmers new tools for integrated pest management. So dear friends, now let us talk about the genetic manipulation of the starter cultures. So starter cultures are the carefully selected group of microorganisms that are intentionally added to milk and milk products to bring a desirable fermentation. The desirable fermentation means, for example, the conversion of milk into dahi by use of lactic cultures. You can say it is a desirable fermentation. So there are number of organisms that comes under the starter culture group, that we can say lactococcus, lactobacillus, streptococcus, leuconostox, and there are number of good bacteria that comes under the starter culture categories. So now what is important in the starter culture? Starter cultures, when they grow, they perform their activity in terms of production of acid and production of flavor, which improves the value of the food product. However, many of the time these features or this activity are lost, or even if we want to modify certain activity of these organisms, we can incorporate certain genes which is the part of the genetic engineering. So in this case, what happened in starter cultures, there is a one molecule, DNA molecule, which is present. We call it as a plasmid. This plasmid is the extra chromosomal DNA, and which is having all types of traits, all type of properties or activities that which bacteria can perform in a positive way. So with the help of this genetic engineering, we can play with this plasmid. That means the useful properties we can transfer from one starter organism to another. So the useful properties are which one? These include lactose and citrate utilization, proteolytic activity, and there are many more activities that we can control with the genetic engineering. So that means we can modify the starter strain. One of the important aspect in the genetic manipulation is the use of plasmid. Plasmid integration vector can also be used to explore and help characterize chromosomal DNA from lactic acid bacteria. Indeed, proteinases and fast resistant determinants have been successfully integrated and stabilized in lactococcal chromosomes, where they are amenable to substantial amplification. The most of the work has been done on the lactococcal species, because these are the very simplest organism whose properties can be easily modified. So by use of genetic manipulation methods, we can not only modify the uh, production certain useful properties, but also we can improve the starter function performance, and also increase the certain the unavailable traits which are positive to the food industries. In food grade biopreservatives, the which is one of the potential area of biotechnology, that is the use of biologically produced molecules, that is we call it as a biopreservatives. So there are certain organisms which can produce molecules which will be helpful to control various spoilage causing organisms. So we can categorize them into the various microbial inhibition, for example bacterial inhibition. In these, the suppression of spoilage on foodborne pathogens by lactic acid bacteria could be extremely beneficial to the human health and dairy industry, as these attributes can considerably improve the shelf life and safety of the fermented foods. In bacterial inhibition, bacteriocins appear to be the most promising candidate for exploring the preservative potential of these bacteria on a large scale. Much of the work being applied to bacteriocins as is often based on approaches and protocols originally developed for the analysis of niacin, which has already been given the GRAS status. Under the fungal inhibition, the mold contamination in dairy foods and feed is a serious problem in third world countries like India, where the tropical humid conditions and unhygienic environments are extremely favorable for the growth and proliferation of these types of organisms. Mainly yeast and molds are found on the feed, and they can produce various types of toxin which can cause a potential threat to the humans, as these toxins are major damaging factor for the human system. Apart from causing serious spoilage problems in these commodities, many of the mold can produce various, as I said, various types of toxins. They can have a carcinogenic effect on the human health. Although the mold contamination is controlled by application of some chemical additives, there are serious concerns regarding the use of these unnatural additives in food chain, as many of the chemical contaminants, chemical preservatives, they are not allowed by the regulatory authorities. As a result, there is growing concern of use of certain naturally fungal protecting cultures, that means which can control the fungal spoilage or even the fungal species by producing certain metabolites. The example of which that there are certain molecules which can produce by certain lactobacilli, and which has been shown the antifungal or anti-mold or anti-yeast effect, which can subsequently uh, prevent the spoilage by these fungi. So now let us come about the dairy enzymes. Dairy industries requires large amount of rennet to produce cheese in bulk. Traditionally, rennet is procured from the cows, but there is a lot of concern about the use of calf rennet. So there are some ethical issues also, and some countries have banned the calf rennet. Due to this, there is always demand for the cheese production, but there is a lack of supply of the calf rennet. Due to the global imbalance between production of cheese and calf slaughter and worldwide shortage of calf rennet, exploration of alternative source of rennet was investigated. In certain countries like India, religious feeling has aggravated the need to rennet substitute. That means nowadays there is chances of using the rennet from various sources. Rennet can be obtained from plant sources, animal sources, or even from the microbial sources. So among these, the rennet used from the microbial sources has got a lot of attraction, because microbes are easy to handle, and the rennet produced by microbial species are of good quality. So the production levels are not very high in the producer organism, but their production is not cost effective for commercial exploitation. So by with the use of genetic engineering, enzymes are easier to produce than enzymes isolated from original sources and favored over chemically synthesized substances, because they do not create any products or of flavors in a foods. So this means that if you modify the microorganisms to produce the recombinant enzymes, that can give much better result. One of the examples is recombinant chymosin. Chymosin is also it's an enzyme that is called as rennet, which is generally used during the cheese making. In recombinant chymosin and other dairy enzymes, India's cheese industry is expanding rapidly, and hence the demand for calf rennets or its substitute is also showing a phenomenal increase. However, in India, the cheese made with the help of calf rennet is not acceptable to the large sector of consumers due to the religious sentiments. Many of the people do not want the rennet, which is obtained from the calf after slaughtering the calf, which may create the ethical issues, which may raise the ethical issues. Hence, for the large scale production of uh, rennet enzyme, so people have used biotechnological tool or genetic engineering to produce higher quantity of rennet. So by cloning the genes of interest from buffalo calves in E. coli and other hosts, it is now possible to produce recombinant buffalo chymosin for commercial use in cheese industry. As we know that for preparation of cheese, rennet is a very essential enzyme, because for coagulation of milk, certain enzymes are essential, and rennet are the acid proteinases which act on the protein molecule, especially on the kappa casein, which leads ultimately leads to the formation of firm coagulum. The DNA of calf chymosin has been successfully cloned into yeast. The species Chloromyces lactis bacteria includes E. coli and molds Aspergillus niger. The treatment of milk with the galactosidase results in the hydrolysis of lactose, therefore making it digestible by lactose intolerant people. As we know that the people, those who are lactose intolerant, they cannot utilize lactose, and as a result they cannot digest the milk, and the milk will as such pass through the system. So people with lactose intolerant will have more problem. So for this reason people are using various type of supplements where the lactose can be easily absorbed or utilized. So alternatively there are the biotechnological tools by which we can use the enzyme beta galactosidase, which can produce by the biotechnological techniques from certain microbial cultures like yeast, for example Chloromyces lactis and Chloromyces maxicinus, and molds such as Aspergillus niger and Aspergillus oryzae. Another use of biotechnology that proteases and lipases can be used in the cheese industry for flavor development, whereas the beta galactosidase can find application in the production of lactose-free milk intended for lactose intolerant patients. In accelerated cheese ripening, so we know the problem of cheese ripening, it takes longer time. The time requires for ripening or maturation of cheese varies from 3 months to 1 year. So this is a huge time which not only takes the time but also involves a lot of cost, which will create burden on the manufacturer. Alternatively, with the use of certain biotechnological tools, we can manufacture or produce a genetically engineered enzymes which further can increase or shorten the cheese ripening time. Modified or genetically tailored microorganisms and enzymes are now being used for enhancing flavor production in cheese, especially lipases, which have a major role in flavor generation. So lipases can be incorporated in the cheese, where we can again improve the flavor at the lesser time. Enzyme addition is now one of the few preferred methods of accelerated ripening of cheese. Enzymes can be incorporated in the cheese in different forms like immobilized or encapsulated, where we can produce the product in a quick time with uniformity. Nowadays, these approaches are being adopted so that the cheese can be produced in a controlled manner at faster rate. Now, in the rapid detection of food pathogens, we know that high-risk food pathogens such as E. coli O157H7, Listeria monocytogenes, Salmonella shigella, etc., which can gain access into the food due to the mishandling during the production and processing. There are always risk when you are mishandling the product or processes. This kind of pathogens can come into the food chain or food product and may cause a serious health hazard to the human being. There are various methods, including conventional one, which can be useful to diagnose this kind of food pathogens, but these methods are laborious, lengthy and tedious. They do not serve any useful purpose to the dairy industry, as by the time results are available, the concerned food lot is already sold in the market, and hence no follow-up action is possible to avoid the possible outbreak of diseases associated with such a food pathogen. So there is need to develop a simple or fast innovative techniques for rapid detection of pathogens in dairy foods. So there are various types of, nowadays various approaches are available by which we can diagnose the pathogens which are very dangerous even in a within a 30 minutes or 40 minutes. So under the PCR based detection of food pathogens, so we know that under the various molecular techniques, PCR is the simplest of all. The crucial features of this method involve sample preparation, especially a food sample we can take. That food sample is we have to culture, and then we can isolate a particularly target DNA, where in this PCR method there is high reproducibility and greater efficiency. So construction of the actual PCR assays requires nucleotide sequences information and knowledge about the chosen target gene to guarantee maximum specificity and sensitivity. By using different formats of PCR, it is now possible to simultaneously detect more than one food pathogens in the food. For example, if the E. coli, S. aureus or Salmonella are present in the food system, it is possible to diagnose all these three by using a single PCR. PCR-based detection of food pathogens has completely revolutionized the area of diagnostic, as the results are available in less than 24 hours to enable the food processing unit. So as we know that many of the conventional method takes more than 48 hours or even 5 days, 6 days, up to 10 days to diagnose most dangerous pathogens. The PCR based methods are very simple and easier and even faster, that can give results in a day. Now let us come to the RAPD technique. It is called as a randomly amplified polymorphic DNA. In this RAPD assay, also referred to as arbitrary prime PCR, patterns are generated by the application of random DNA segments with a single shot. They are typically a 10 base pair nucleotide primers of arbitrary nucleotide sequence. The primers is not targeted to amplify any specific bacterial sequences and will hybridize under low stringency conditions at multiple random chromosome locations and initiate DNA synthesis. After the separation of the amplified DNA fragments by agarogel electrophoresis, a pattern of bands results, which is a characteristic of the particular bacterial strain. The advantages of these methods are which require a very less time and provides a good discrimination, so we can differentiate even a closely related species of strains, and it can be useful to identify a large number of strains. Under the species-specific PCR, as the name indicates, this type of PCR reaction makes use of species-specific primers for differentiation of different species. Primer sequences usually chosen from highly conserved 16S rRNA and 23S rRNA or 16S 23S intergenic spacer regions. The advantages of this generic region is that all these are a part of highly conserved RNA operon, which has remained well conserved during evaluation and is common to all eubacteria. Moreover, these also contain highly variable regions unique to each particular species. So this means that these regions are highly conserved and which may not find in other species. So all these primers are being used for the identification of different microbial species. Although this is a highly specific technique for species level differentiation, but strain typing is not possible through this methodology. Only we can identify the species, but we cannot go up to the typing level. That means each nucleotide level we cannot understand. However, a straight sequencing of these amplified 16S or 23S intergenic spacer regions in different isolates can easily differentiate even very much closely or even similar strains, and is readily becoming popular among the researchers. So in the next, our methods, which is also called ADRA, amplification and restriction analysis of 16S rRNA gene. Among the other 16S RNA based techniques, ADRA combines both PCR and restriction analysis, and hence is a powerful technique and is capable of differentiating the species as well as strains. Under this method, the restriction endonucleases are used, which are the specific enzyme which can cut the DNA molecules at the specific region. So in this, as we have told, the PCR and restriction analysis PCR method which are combined, which will give a better result. However, since it also has limitations of tedious handling steps, it cannot be applied for large number of samples simultaneously. Coming to the genetically engineered milk, or the humanization of bovine milk, as we know that the breast milk is nature's perfect food for human infants, providing them all aspects of nutrition and protection against the infection. We have heard a lot of time that the mother's milk is the best for the baby. If you try to utilize, use or give any milk to the babies, the baby cannot adapt, or it cannot digest that milk because of the differences in their composition of the milk. There are various attempts have been made to improvise the bovine milk which can be given to the small babies or infants. So number of infant formulas have been manufactured and tried. These formulations can contain major compositional changes, especially proteins. As we know that the mother's milk is very less in protein. So if we give a more amount of protein in the bovine milk, that baby cannot digest the milk. So for this reason, we have to manipulate the protein quantity or composition. We have to lower down the protein composition, which can be easily digested by the infant or baby. In this regard, even people have tried to change various proteins or their enzymes. The composition of these infant formulations can be also improved if the proteins contained therein more closely resemble those of human milk. It is now possible to add the human lactoferrin or lysozyme to the bovine milk by genetic engineering to produce new functional foods. There is always possibility of incorporating many number of proteins or enzymes with the help of biotechnological tools, which will not only improve the value of the product, but also it will be very helpful for the baby's growth. The shelf life of such product is also expected to be very high due to the antimicrobial activity of these proteins. As we know that lactoferrin, or just to cite an example, lactoferrin, which is an enzyme which have shown anti-microbial effect, because lactoferrin is an enzyme which bind to the iron, and we know that there are number of organisms which requires iron for their growth, and if this enzyme binds the iron, the free iron is not available for the microbial activity. As a result, the microorganisms get inhibited. The goal of the dairy industry has been to create an efficient, healthy cow buffalo that can serve all the needs of the industry. Genetic engineering offers the opportunity for a paradigm shift, a reshaping of the industries from the producers to the processing plants. Dairy producers have the opportunity to choose to produce high protein milk destined for cheese manufacture that has accelerated curd clotting time, milk containing nutraceuticals, orally administered biologicals that provide health benefits or replacement for infant formula. Such a scenario would be a radically change for the dairy industry. There is now a little doubt that the products of the genetic engineering will become a part of the dairy industry in the upcoming days. Now coming to the modified milk in transgenic dairy cattle have only one copy of the genes that encode alpha S1 oblique S2, beta and kappa casein proteins, and out of the four caseins, kappa and beta are the most important. We also know that the kappa and beta casein are very important as far as cheese making is concerned. So in this case also, these two proteins are very important. Increased milk casein content reduces the size of the micelles, resulting in improving the heat stability. Heat stability has been correlated with the casein. Beta caseins are the highly phosphorylated and bind to the calcium phosphate, thus influencing the milk calcium levels. Research on modification of the milk composition to improve nutritional or functional properties has been mostly done in the transgenic mice. The transgenic cells become the donor cells in the process of nuclear transfer, generating 9 fully healthy and functional cows. There has been research that the improvised, or we can say the transgenic animals have been produced with the very good results. Overexpression of uh, CSN2 and CSN2 oblique 3 in the transgenic cows, that means cow caseins, resulted in an 8 to 20% increase in beta casein and 100% increase in kappa casein levels. And now the next topic is the elimination of carcinogenic compounds from the food. There is a very good example of brewer's yeast, that is Saccharomyces cerevisiae, is one of the most important and widely used microorganisms in the food industry. Especially these Saccharomyces cerevisiae are used during the manufacture of wine, beers, and other beverages. The recombinant DNA technologies has made possible to introduce a new properties into the yeast as well as to eliminate undesirable byproducts. One of the undesirable byproducts formed during the yeast fermentation of foods and beverages is the ethyl carbamate or urethane, which is a potential carcinogenic substance which can cause cancer in the susceptible humans. Ethyl carbamate is synthesized by the spontaneous reaction between ethanol and urea, which is produced from the degradation of arginine found in large amount in grapes. Yeast used in wine fermentation possesses the enzyme arginase that catalyzes degradation of arginine. If these enzymes can be blocked, arginine will no longer be degraded into urea, which in turn will not react with the ethanol to form ethyl carbamate. So now coming to the most catching area, that is the catching area of biotechnology, is the use of proteomics. In the application, the next topic is the application of proteomics in the food and dairy sector. Proteomics technology is routinely used, and the terms industrial process proteomics and industrial proteomics are now frequently used. Proteomics are the tool, or is the tool which is used to identify various types of proteins which are produced during the process. For example, certain proteins are produced during the microbial growth or fermentation. That proteins we can even quantify and identify by the use of proteomics techniques. So what proteomics techniques? So there are a set of steps, a set of procedures are involved to identify a particular proteins. So proteomics, which includes the use of gel electrophoresis, then there are non-gel electrophoresis approaches which will not require uh, agar gel electrophoresis, and without agar gel electrophoresis we can identify the particular proteins. These techniques offers a powerful new way to characterize the protein component of the foods. Proteomic techniques offers a way to investigate differences in the protein composition of different tissues within a specific animal or vegetable food type, as well as between different varieties of it. So we know that milk contains complex proteins that undergo several qualitative and quantitative changes during the processing and storage. So we can identify various types of proteins by using proteomics techniques. So there are various tools in that, or various approaches we can use under the proteomics. We can say that there is shotgun approach, or we can say directly we can go for the uh, identifying the spectrum of the particular protein. In the last years, there is a dramatic uh, increase in the use of such a powerful or some most sophisticated techniques. Uh, Among these, the proteomics has got much attention because of the it's a dynamic because this technique is very dynamic and gives a very robust, it's a robust and gives very accurate result, and it has been successfully used in the milk and food system. So now students, you must have understood the role of biotechnology in the food industry. Now let us come towards the summary of this module. Biotechnology as applied to food processing to make use of microbial inoculants to enhance properties such as the taste, aroma, shelf life, texture, and nutritional value of the foods. Biotechnological approaches can be employed for improving productivity, economy, physicochemical, and nutritional attributes of a wide range of food products. As we have seen already, this can be used in many dairy products and other food beverages. Biotechnology based methods can provide accurate results within a relatively short time frame. As compared to conventional methods, biotechnological methods are more reliable and gives results in very short time. Biotechnological developments have resulted in the widespread availability of low-cost rapid methods of identification when compared with the significant cost time requirements of the conventional techniques. Thank you.